All NEET aspirants, very good morning. With regard to botany, the 11th NCRT, the forsake of the NEET, it is my attempt to make your process easier in learning the concepts of NCRT botany. As far as this physiology part concerned, I want to opt this the topic, the respiration. So keep the textbook with you. How my delivery is going to be synchronized with the text, you check it. And certainly it is going to unravel all the twists, the knots in each paragraph. And it makes your process of the learning much easier. Okay? Keep the textbook with you. So respiration in case of the plants, in very first paragraph, the breathing and respiration. Breathing and respiration, the words it is not interchangeably used. If you take syngamy and fertilization, which are interchangeably used. Even though the words breathing and respiration not interchangeably used, but it is interconnected. How it is interconnected? Breathing it is the physical, respiration it is physiological. All we breathe to live. What happens when we breathe? We inhale oxygen. The blood carries oxygen to various vital organs required for vital organs as it provides energy by oxidation of food. So respiration it is physiological, breathing it is physical. Do all organisms breathe? Yes. Plants, microbes, animal, all breathe with or without breathing organs. Next, we require energy to do every activity. The movement, the transport, absorption, reproduction, even for breathing, we require energy. Where do we get this energy? From food. How the energy is taken from the food? By oxidation of the food, aerobic or anaerobic respiration. How this energy is stored? In a chemical bond. In which form? It is ATP. Do all foods having the same amount of energy? Not at all. The fats, it is high energy rich. The carbohydrate is less than the fat, but it is instantaneous energy rich. So all foods do not have equal amount of energies. Next, do plants eat for sake of energy? Not at all. Plants for sake of energy depends upon the sunlight. It makes the food. Do microbes eat? Microbes never eat. For sake of energy depends upon the sunlight in case of photoautotrophs. In case of oxidation of inorganic substrate, that is chemoautotrophs. Organic substrate for sake of the carbon, for sake of energy, it depends upon the organic source. What do you call? Chemoheterotrophs. Next. So all the questions arising with regard to the breathing and respiration not seems to be connected. But in reality, the breathing requires energy, that energy provided by oxidation of food in respiration. Whatever the macromolecule that is required for oxidation to provide energy is obtained by a process photosynthesis. The green plants and blue-green algae which convert light energy into chemical energy and stored in CC bond, the sugar, hexose, starch, sucrose. So the plants which prepare food material by placing carbon dioxide and water in presence of the light, what do you call? Autotrophs. So green plants, all cells which make the food are green in color? Not at all. The cells lie superficially are green in color which makes the food. What about the cells, the tissues, organs which are non-green? Food needs to be translocated to those parts in order to oxidize the food to release energy. So food prepared in the green cell, it translocated to non-green cells. And all organisms depend upon the plants, the autotrophs, directly or indirectly, what do you call? Heterotrophs. Maybe it is herbivores and the carnivores. Directly it is herbivores, indirectly it is carnivores. Next, the saprophytic fungi, saprophytic bacteria depends upon dead decaying organic matter. The food that is oxidized to release considerable amount of energy in a cell, what we call cellular respiration. In next paragraph information, undoubtedly photosynthesis occurs in chloroplast, light phase in grana, dark phase in stroma. Whereas respiration in case of the prokaryote that occurs in cytoplasm as well as mesosomes which are involved. In case of eukaryote, it occurs in cytoplasm as well as mitochondria. And whatever the food organic substance that oxidizes to release energy, what we call respiratory substrate. Common respiratory substrate in all organisms is carbohydrate. In some plant, in some circumstances, fats, protein, organic acid are also respiratory substrate. And these respiratory substrate oxidizes to yield energy. And the breakdown of the substrate is not in a single step, not directly released, energy not directly released into the cell. And energy not directly it is utilized in many small steps and energy packed in the form of ATP. So energy trapped in ATP. And this ATP is considered as energy currency of the cell. Why? Because it is available wherever, whenever it is required. That's why it is energy currency of the cell. Powerhouse of the cell, ATP production, it is mitochondria. It is energy currency of the cell. And what are the aspects present in case of ATP? Adenosine triphosphate. What type of nitrogen base? Purine nitrogen base, adenine. It is heterocyclic compound, dicyclic compound. And sugar, ribose sugar, that is the pentose, 5-carbon sugar. And 3-phosphate. So it is a ribonucleotide. Otherwise it is ribonucleoside triphosphate or ribonucleotide diphosphate. Next, the carbon skeleton, what we call it is intermediates formed in this process of oxidation, which are utilized to synthesize other biomolecules. How it is withdrawn when the process is going on, as the process occurs very slowly in many small steps. So as it is slow, the substrates, intermediates are withdrawn to synthesize other biomolecule needed by the cell. Next heading, do plants breathe? The heading. Answer, undoubtedly it is not so direct. Because in contrast to animal, plant do not have any breathing or respiratory organ. But it ensures the supply of carbon dioxide and oxygen through stomata in younger part, lenticels in older parts. Next, how plant can get along even without respiratory organs. It mentioned in 3 aspects, first, second, third. In first aspects, there are 2 key points. The first key point in first aspect, each part of the plant can take care of its own needs. Second key point in first aspect, there is little transport of the gases from one part to other. In second aspect, the first key point, plant do not present great gaseous exchanges demand. Root, stem, leaf respires far lower than animals. That's the second key point. Third, much gas exchange occur only during the process of photosynthesis. Next key point, leaf is well adapted to take care of the needs during this period. Why? Oxygen availability is not a problem during these periods of photosynthesis. Third aspect. In third aspect, the key points which are, even in a large bulky tree, the distance that the gases that diffuse is not that much of great. Next key point. Most of the living cells lies close to the surface. That is true in case of the leaf. It is contact with atmosphere. What about thick woody stems and root? If you take thick woody stem, thin layers of the cell beneath the bark. What is thin layers of the cells beneath the bark? Secondary cortex. It do have the pores. What are the pores? Lenticels. Much interior provide mechanical support. What is much interior? Secondary xylem. That is the wood that provide mechanical support. And last point in the third aspect, it is most of the cells, at least a part, contact with atmosphere. How? That is facilitated by loose packaging of cells interconnected by air spaces, leaf, stem and root. How leaf got connected to atmosphere? Through intercellular space of the mesophyll and substromatal cavity to atmosphere. In case of the stem, that is intercellular spaces of the cortex. In case of the root, intercellular space of the cortex, general cortex. So root communicated to atmosphere through stem as well as leaf. So most of the cells, at least a part, contact with atmosphere. Suppose it is assertion. The reason it is facilitated by loose packaging of the cells interconnected by air spaces. Next, if the released energy is really useful to the cell to synthesize other biomolecules needed by the cell adopt a clever strategy. The key is the glucose catabolize in such a way that the released energy not completely goes out as heat. Energy not directly released into the cell. Not in a single step. Many steps, small steps, some steps large enough to make more number of ATP, like it is electron transport system. As the process occurs very slowly, the substrates are withdrawn as and when required to synthesize other biomolecule needed by the cell. So it is nothing but entire story of respiration. In next paragraph, the products of respiration: carbon dioxide, water as well as energy. And there are many evidences that first organisms or first cells appeared survived under anaerobic condition. Can you see any organism or situation or instances? Yes. If you take facultative anaerobe, originally it is aerobe. It survives even under anaerobic condition. If you take obligate anaerobe, survives only in absence of oxygen. Obligate anaerobe. In presence of oxygen, it cannot survive. Can you see the situation? Yes. In if you take the seed germination, initial seed germination, it is anaerobic. Later it becomes aerobic. Deeper tissues perform anaerobic respiration only. Whether it is aerobic or anaerobic, all organisms retain enzymatic machinery to partially oxidize glucose into two molecules of pyruvic acid in cytoplasm. What do you call? Glycolysis. So glycolysis is only the process in case of anaerobes to yield energy. So whether aerobic respiration or anaerobic respiration, glycolysis is the common pathway. Next, the sucrose, the end product of the photosynthesis, is a disaccharide. In presence of invertase enzyme, is hydrolyzed into glucose and fructose to monosaccharide. And the glucose and fructose monosaccharide readily phosphorylated and enter into this glycolytic pathway. So glucose is a simple substrate that directly participates in respiration. Next, in aerobic respiration, there are 6 oxidation. None of the reaction there is no involvement of oxygen. Removal of redox equivalent hydrogen from respiratory substrate, what do you call it, is oxidation. Total oxidations in aerobic respiration 6. Glycolysis 1. In between glycolysis and Krebs cycle, that is link reaction, it is 1. Krebs cycle 4. So total it is 6 oxidations. So first it is glycolysis. It occurs in cytoplasm as it was discovered by Embeden, Meyerhoff, Paranas. So it is called EMP pathway. So glycolysis is enzymatic breakdown of the glucose into two molecules of pyruvic acid in cytoplasm. What do you call it is glycolysis. One molecule of glucose. Finally, it is going to yield two molecules of pyruvic acid. In this, there are total 10 reactions, except 1, 3, 10, all which are reversible reactions. The 1 to 10 reaction: phosphorylation, isomerization, phosphorylation, cleavage, isomerization, oxidation, dephosphorylation, intramolecular shift or isomerization, dehydration, dephosphorylation. In first step, phosphorylation, glucose is phosphorylated in presence of hexokinase by utilizing ATP and converted into glucose 6-phosphate and ADP. So in first step, ATP is utilized. In next step, glucose 6-phosphate is isomerized to fructose 6-phosphate, because in next step is going to utilize ATP. So fructose 6-phosphate is only useful in phosphorylation step. That's why glucose 6-phosphate is isomerized to fructose 6-phosphate in presence of hexose isomerase. Fructose 6-phosphate is phosphorylated in presence of phosphofructokinase by utilizing ATP, converted into fructose 1,6-biphosphate. The fructose 1,6-biphosphate is 6-carbon compound. The first carbon having phosphate, sixth carbon also having the phosphate. Next, fructose 1,6-biphosphate in presence of aldolase, it cleaved into two trioses, phosphorylated trioses, glyceraldehyde 3-phosphate or 3-phosphoglyceraldehyde and dihydroxyacetone phosphate. How two phosphorylated trioses are formed? Because in earlier step, fructose 6-phosphate phosphorylated to form fructose 1,6-biphosphate. That's why two phosphorylated trioses are formed by cleavage reaction. So by cleavage reaction, what products are formed? G3P and DHAP. In case of C3 pathway, G3P or PGL, that is formed by reduction process. Here it is the cleavage process. Next, in next step of the catabolism, DHAP is not useful. It is converted into, again it is the G3P in presence of triose phosphate isomerase. So how many G3P molecules are formed? 2 G3P molecules are formed. In sixth reaction, glyceraldehyde 3-phosphate undergoes oxidation. So redox equivalents are removed, hydrogens which are removed from G3P. From 1 G3P, 2 hydrogens are removed. From 2 G3P, 4 hydrogens are removed. And these hydrogens which are added to NAD got reduced to NADH2. And there is utilization of cytosolic phosphate. So already it is glyceraldehyde 3-phosphate. With addition of cytosolic phosphate, it is going to become 1,3-biphosphoglyceric acid. First carbon having phosphate, third carbon also having phosphate, 1,3-biphosphoglyceric acid in presence of triose phosphate dehydrogenase or glyceraldehyde 3-phosphate dehydrogenase. So sixth reaction, it is oxidation. So first time in aerobic respiration, which compound undergoes oxidation? Glyceraldehyde 3-phosphate. The first oxidation product is 1,3-biphosphoglyceric acid. Next, 1,3-biphosphoglyceric acid undergoes dephosphorylation. So phosphate from the first carbon readily removed and added to ADP. ADP is going to become ATP in presence of, that is, kinase enzyme. So 1,3-biphosphoglyceric acid is converted into 3-phosphoglyceric acid with the formation of ATP. This is the first substrate level phosphorylation, 1 ATP is formed. So, what is the substrate for first substrate level phosphorylation? 1,3 biphosphoglyceric acid. Next, 3-phosphoglyceric acid is isomerized to 2-phosphoglyceric acid. It is an intramolecular shift. The phosphate present on the third carbon, it got shifted to second carbon. What do you call it? Is intramolecular shift. 2-phosphoglyceric acid. Next, the 2-phosphoglyceric acid in presence of enolase undergoes dehydration to form phosphoenol pyruvic acid. So this phosphoenol pyruvic acid in glycolysis is formed by which reaction? Dehydration. The same PAP in case of C3 plant mesophyll cell formed by phosphorylation reaction. Pyruvic acid by utilizing ATP phosphorylated to become phosphoenol pyruvic acid. In this it is formed by dehydration reaction. Next, phosphoenol pyruvic acid undergoes dephosphorylation as the phosphate is removed and added to ADP. Phosphoenol pyruvic acid become pyruvic acid with the formation of 1 ATP. So this is second substrate level phosphorylation in glycolysis. The substrate for second substrate level phosphorylation is PEP and product it is pyruvic acid. So for 1G3P, 2 ATP are formed. One at 1,3,2,3 phosphoglyceric acid, the second at phosphoenol pyruvic acid to pyruvic acid. For another G3P, the 2 ATP are formed. So total ATP formed in glycolysis for 1 fructose 1,6 biphosphate or 1 glucose or 2 G3P is 4. But in first and third reaction, there is utilization of the 2 ATP. So net ATP is going to become 2 ATP. So total number of ATP 4. Net gain of ATP 2. Number of pyruvic acid molecules formed 2 pyruvic acid molecule. Number of redox equivalents which are removed 4 hydrogen. For 1 G3P2, another G3P2, 4 hydrogen. Number of NADS2 formed 2 NADS2 formed. There is no utilization of oxygen. There is no evolution of carbon dioxide. So glucose is the substrate. The total ATP formed 4. Net gain it is 2. If glucose 6-phosphate is the substrate, total ATP is going to be 4. But net gain is going to be 3. If fructose 6-phosphate, again the same answer. The 4 ATP, total 3 ATP net gain. And fructose 1,6 biphosphate, there is a total of 40 is the 4 ATP. There is no utilization. It is a hypothetical question. If one molecule of fructose 1,6 biphosphate directly participate in respiration, total ATP is going to yield 4 ATP. So ultimately the pyruvic acid is formed at the end of glycolysis. And the metabolic fate of the pyruvate, it depends upon cellular needs, prevailing condition, whether aerobic or anaerobic, and organism, whether aerobic organism or anaerobic organism. So metabolic fate depend upon these conditions, type of organism, prevailing condition. So metabolic fate depends upon these conditions, and the different cells handle the pyruvic acid differently. After glycolysis, it may be alcoholic fermentation, lactic acid fermentation, aerobic respiration. In case of fermentation, anaerobic breakdown of the glucose in most of prokaryote and unicellular eukaryote, anaerobic breakdown of glucose. In case of alcoholic fermentation, pyruvic acid undergoes decarboxylation in presence of pyruvic decarboxylase to form acetaldehyde. So which undergoes decarboxylation? Pyruvic acid. Decarboxylation product it is acetaldehyde. Acetaldehyde reduced to ethyl alcohol. The redox equivalents for reduction of acetaldehyde are provided directly by NADH2. So ultimate hydrogen donor for this reduction process of acetaldehyde to ethyl alcohol is glyceraldehyde 3-phosphate. Immediate hydrogen donor for reduction of acetaldehyde is NADH2. Ultimately, alcohol is formed and energy. So co-product CO2, finally it is ethyl alcohol. So in these questions, which undergoes decarboxylation? Pyruvic acid. Which undergoes reduction? Acetaldehyde. Which undergoes oxidation? NADH2. NADH2 oxidized by acetaldehyde. And final product it is ethyl alcohol. Immediate hydrogen donor to acetaldehyde, NADH2. Ultimate hydrogen donor, G3P. Next, lactic acid fermentation. In lactic acid fermentation, in presence of enzyme lactate dehydrogenase in bacteria, lactobacillus, the pyruvic acid undergoes reduction to form lactic acid. The two NADH2 are utilized in the formation of lactic acid. Only it yield energy, there is no carbon dioxide. So common thing for alcoholic fermentation and lactic acid fermentation is energy. There is no evolution of CO2 in lactic acid fermentation, evolution of CO2 in alcoholic fermentation. The questions in case of the lactic acid fermentation. Which undergoes reduction? Pyruvic acid. Which undergoes oxidation? NADH2. The product formed? Lactic acid. NADH2 oxidized by pyruvic acid. In skeletal muscle cell, during strenuous exercise, there is inadequate supply of oxygen in case of the muscle cells. During that period, what is formed? Lactic acid is formed. Whether alcohol or lactic acid is hazardous, that is damaging to the cell. And in case of yeast which performing fermentation, yeast poison themselves to death, about 13% alcohol. So in natural alcoholic fermentation, alcoholic concentration is only 13%, because there is no further production of the gymase from the yeast cell, because yeast poison themselves to death by 13% of alcohol. So there is no further increment. So percent of alcohol in natural beverages obtained in fermentation process is 13%. What is the reason for high alcoholic concentration in the beverages, other beverages obtained in the market, which are obtained through distillation process? It is having high alcoholic content. If you take the wine and beer, with it is without distillation, having low alcoholic content. The vodka, brandy and gin, which is having high alcoholic concentration, obtained through distillation process. Next, in anaerobic respiration, less than 7% of energy is released from the substrate and not all trapped in ATP. So net gain of ATP in alcoholic fermentation or lactic acid fermentation is only 2 ATP. Whatever the number of ATP obtained in glycolysis is only the net gain in case of alcoholic fermentation. So net gain of ATP is the 2. In skeletal muscle cells under aerobic condition, what is formed? Pyruvic acid is formed. And skeletal muscle cells under anaerobic condition, what is formed? Lactic acid is formed. Next. In aerobic respiration, the pyruvic acid undergoes complete oxidation to carbon dioxide and water with release of more energy that trapped in more number of ATP. So complete oxidation of the food substrate in case of aerobic respiration with a large number of particles ATP. So in alcoholic fermentation, lactic acid fermentation and aerobic respiration, NADH2 undergoes oxidation. In alcoholic fermentation, NADH2 oxidized by acetaldehyde. In lactic acid fermentation, NADH2 oxidized by pyruvic acid. In aerobic respiration, NADH2 oxidized by oxygen. So oxidation of NADH2 is slow in alcoholic fermentation and lactic acid fermentation, as it is oxidized by acetaldehyde and pyruvic acid. In case of aerobic respiration, oxidation of NADH2 is vigorous, very fast, because it is oxidized by oxygen. Next, in heading the aerobic respiration. The aerobic respiration, there is involvement of mitochondria with a large number of ATP form. You see the heading, the aerobic respiration, the first and 2 points. Removal of all hydrogens from pyruvic acid, leaving 3 molecules of CO2 that occur in mitochondrial matrix. That is one point. The second point is passing on of electron removed as the part of hydrogen transferred to oxygen with simultaneous synthesis of ATP. So passing on of electron removed as the part of hydrogen transferred to oxygen with simultaneous synthesis of ATP. What do you call the process? Oxidative phosphorylation, where it occurs inner mitochondrial membrane. So one process that occurs in mitochondrial matrix and the second process that occurs in case of inner mitochondrial membrane. So these are the aspects up to this. Then we are going to cover this aerobic respiration. So pyruvic acid formed in glycolysis in cytoplasm, it enters into the mitochondrial matrix for further oxidation that will cover in case of aerobic respiration events. In this oral coverage, which substrate participate in respiration directly? Glucose. Which undergoes oxidation first time? G3P. First oxidation product is 1,3 biphosphoglyceric acid. Which is the substrate for substrate level phosphorylation? 1,3 biphosphoglyceric acid. Product it is 3-phosphoglyceric acid. What is the substrate for second substrate level phosphorylation? Phosphoenol pyruvic acid. What is the product? That is the pyruvic acid. So number of ATP formed in glycolysis 4. Net gain of ATP is the 2. Number of pyruvic acid molecule 2. Number of NADs formed 2. Number of redox equivalents removed for 1 glucose in glycolysis the 4.